Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
We often think about our genes, how they're controlled, as being pretty fixed. Like you flip an off switch for a gene and that's it. Maybe it even gets locked down tighter over time. But what if one of the most important gatekeepers for locking things down, when we thought only added locks, actually had a secret way to take them off? There's this brand new paper in Science that's just turned that whole idea upside down.
Yeah.
It's found this hidden master control for silencing parts of our DNA. And that's the fascinating puzzle we're diving into today.
Precisely. Yeah, this deep dive is all about a really fundamental epigenetic mark. It's called H3K9me3. Now, for years, we've had a decent handle on how this silencing mark gets, onto our DNA and how it's maintained. It involves this thing called a positive feedback loop. You can sort of picture it like a snowball rolling downhill, right? It just gets bigger and bigger, makes the silence stronger.
Okay, yeah. More silence, more lockdown.
Exactly. But this really elegant self-reinforcing system, it created a huge puzzle for scientists. If it is just positive feedback, why doesn't this silencing just spread everywhere? Why doesn't it just paint the whole genome silent and, completely stop the cell from working? I mean, that would prevent life itself.
Right, it can't just be an accelerator. There has to be a brake somewhere.
That's it. And this new research, it finally unveils this crucial negative regulator, the brake we've been looking for. It keeps everything in balance, makes sure epigenetic inheritance works properly, and stops this runaway silencing, this excessive heterochromatin formation.
Okay, before we get to the big reveal, maybe we should just quickly make sure we're all clear on what these off switches actually are. Could you give us a quick rundown of H3K9me3 and heterochromatin, like the master dimmer switch analogy you mentioned? Why are they so important?
Absolutely. So imagine our DNA, right? Your entire genetic blueprint. Think of it like a giant library. Now, some sections in this library are packed incredibly tightly. They're sort of locked down. We call these sections heterochromatin. And the genes inside these tightly packed regions are mostly silent, like books stored away in a restricted area you can't easily access.
Okay, so heterochromatin is the closed-off section of the library.
Exactly. And H3K9me3, or histone H3 lysine 9 trimethylation if you want the full name, that's basically like a little chemical tag, a sticky note, on the histone proteins. Remember, DNA is wound around these proteins called histones, and these H3K9e3 sticky notes are the signal saying, keep this region tightly closed, keep it silent.
Got it. Sticky notes for silence.
Yep. Now, the classic model for how this silence gets maintained, it involved this right and red positive feedback loop. It's actually quite elegant. You have a protein called HP1, that's the reader. It recognizes these H3K9me3 sticky notes.
Okay, reader finds the note.
Right. And once HP1 binds, it then recruits a different protein, an enzyme called a methyltransferase. Specifically, something like SUV39H1. You can think of SUV39H1 as the writer. Its job is to add more of these H3K9E3 sticky notes to the histones nearby.
So the reader calls over a writer to add more notes, reinforcing the silence.
Precisely. It reinforces the silence, makes the heterochromatin even tighter. It's a really efficient system for keeping silent bits silent. But like we said, this positive feedback, it created that central paradox. If it just keeps adding marks, how do cells stop it? How do they prevent this silencing from just spreading like wildfire across the whole genome, maybe accidentally switching off essential genes the cell desperately needs?
Yeah, that seems like a massive potential problem.
It is, and this is exactly where the new paper's findings come in. They found the fire extinguisher, so to speak.
Okay, so they knew the problem, this runaway sticky note situation. The big question then is, how did they actually find the solution? What was their approach to discovering the break? Because you mentioned the methods were pretty cutting edge too, right? That's part of the story here.
Oh, absolutely. Their research strategy was really clever. They started with a hypothesis. They reasoned there had to be some kind of active mechanism, not just, a lack of writing, but an active removal or maybe degradation. And they suspected it might involve a specific type of cellular machinery, ubiquitin ligases.
Ubiquitin ligases.
Oh.
Those are like the cell's cleanup crews, right? Tagging proteins for disposal.
Exactly. They tag proteins for destruction by the proteasome, the cell's recycling center, prevents junk from piling up. So, to find this potential regulator, they launched this huge, unbiased, genome-scale CRISPR-Cas9 knockout screen. Think about that they systematically knocked out or disabled almost every single gene in the human genome, one by one, in different pools of cells to see what happened.
Wow, okay. That's a massive undertaking. How did they track the effect on silencing?
That's the other clever bit. They used these specially engineered reporter cells. These cells had a built-in fluorescent switch, basically. This switch was designed to light up, to glow brightly, only if H3K9me3 levels went up too high, if the silencing got out of control. So the logic was, if they knocked out a gene that normally acts as a brake on H3K9me3, then removing that brake would cause H3K9me3 to pile up, and the cell would light up.
You got it. Without that negative regulator, H3K9me3 accumulates, and boom, the reporter glows. So they screened something like 19,000 genes, a monumental effort. And from all that data, they pinpointed one specific complex, the cellular cleanup crew, the CUL5-ASB7 E3 ubiquitin ligase complex. That was the break.
CUL5-ASB7, okay.
And this wasn't just luck. This combination of a quantitative fluorescence assay with deep sequencing. It's a real step change in how we find these epigenetic layers. Much faster and more comprehensive than older methods. Okay, so they found ASB7, the big reveal. The break exists. Now, the next question is obvious. How does it work? What exactly does this ASB7 complex do to keep H3K9me3 in check? Does it, scrape off the sticky notes? Or is it doing something else to stop that wildfire?
Yeah, good question. Once found, what's the mechanism? So they dug into that using multi-omics analyses. That just means they looked at lots of different things simultaneously. Gene expression, protein levels, where proteins were binding on the DNA all across the genome, and the pattern was crystal clear. When you get rid of ASB7, when you remove that break.
H3K9me3 goes way up.
Way up. It hyperaccumulated across the genome, and this caused more HP1, the reader protein, remember, to bind everywhere. It even started silencing genes near repetitive DNA elements, genes that probably should have stayed active or at least flexible. The silencing just spread too far.
Okay, so loss of ASB7 means too much silencing.
Yeah.
How does ASB7 normally prevent that?
Right, the mechanism. This is where it gets really interesting. They found that ASB7 is actually recruited to these silenced heterochromatin regions by HP1 itself.
Wait, so the reader protein HP1 actually calls over the break ASB7. That seems counterintuitive.
It does a bit, doesn't it? But it's like HP1 is part of a built-in control system, inviting its own regulation to keep things balanced. Now, once ASB7 is there, it doesn't directly remove the H3K9me3 sticky notes. Instead, it goes after the writer.
The writer? You mean the methyltransferase like SUV39H1 or others?
Exactly. They showed specifically that ASB7 targets a key methyltransferase called SETB1. What ASB7 does is it ubiquitinates SETB1. It sticks that destroy me tag onto it.
Ah, the ubiquitin tag from the cleanup crew function.
Precisely. That tag marks SETB1 for destruction by the proteasome, the cell's recycler. So it's this really novel erase the writer strategy. Instead of just inhibiting the enzyme that adds the marks, ASB7 actively gets rid of the enzyme itself.
Wow, that's direct. Taking out the source.
It is, and they confirmed this was really happening. They did rescue experiments using versions of ASB7 that were ligase dead, meaning ASB7 was there but couldn't attach the ubiquitin tags, and those dead versions couldn't act as a brake. They even mapped exactly where ASB7 physically connects to SETB1. It's a very specific, very active form of control.
Okay, that's an incredibly elegant mechanism, erase the writer. I like that. But, our cells aren't static. They're dividing all the time. Genes are switching on and off. Life is dynamic. So is this regulation, this balance between writing and erasing, is it constant? Or does ASB7 play a role in the dynamics, especially during something like cell division, mitosis? Because making sure the daughter cells inherit the right silencing patterns seems absolutely critical.
You've hit on a crucial point. The dynamic aspect is key. Think about it. Every time a cell divides, it has to copy its DNA perfectly, right? But it also has to copy its epigenetic state. All those sticky notes, all those on-off switches have to be put back in the right place on both copies of the DNA.
Right. Otherwise, the daughter cells would be completely different.
Exactly. And the study found something really remarkable about ASB7's role during mitosis, that critical phase of cell division. They discovered that during mitosis, another protein called CDK1, which is a major driver of the cell cycle, it actually modifies AST7. It phosphorylates it.
Phosphorylates adds a phosphate group. like a temporary flag.
Sort of like a temporary pause button, yeah. In this case, when CDK1 phosphorylates ASB7, that phosphorylation stops ASB7 from interacting with another key methyltransferase, SUV39H1. Remember, SUV39H1 is another major writer of H3K9me3.
Okay, so during division, ASB7 is temporarily told to back off from degrading SUV39H1.
Exactly. ASB7 is disengaged for a bit, and this allows SUV39H1 to stabilize, to not get degraded during this critical window. And why is that important? Because the stabilized SUV39H1 is then ready and available to quickly and accurately put the H3K9me3 marks back onto the chromosomes right after the cell has finished dividing.
Ah, I see. So pausing the break allows the writer to quickly reestablish the silencing pattern after the DNA has been copied.
Precisely. It reveals this really beautiful dynamic circuit involving HP1, SUV39H1, and ASB7, all working together to control H3K9me3 levels through the cell cycle. And why this matters to you listening is that this precise timing, this regulation, is absolutely essential for what we call faithful epigenetic inheritance. It makes sure your daughter cells get the correct gene expression patterns, and crucially, it prevents that excessive heterochromatin, that uncontrolled silencing, which could really mess things up, leading to genomic instability or diseases like cancer where gene control goes haywire. It's fundamental.
Wow. Okay, so it's not just a static break, it's a dynamic regulator timed perfectly with cell division. That's quite something. Now, this research isn't just fascinating for the biology. You mentioned it also involves some pretty innovative techniques. Can you tell us a bit more about what made the approach here so groundbreaking, and maybe how these discoveries might ripple outwards into other areas of biology or even medicine?
Yeah, definitely. Beyond the core discovery about ASB7, the methodological innovation here is really significant. It's a testament to some really smart scientific thinking. The lead investigator, Zhao Li, had this great idea to fuse a fluorescent protein, EGFP, to specific reporters known to be silenced by H3K9me3 mechanisms. This gave them that real-time quantitative readout we talked about. It's like watching a movie instead of looking at still photos.
Much more informative.
Much more. And the postdoc on the paper, Maria Sanchez, apparently worked incredibly hard to refine a technique called tandem affinity purification. This was needed to capture the very brief, very transient interactions between ASB7, the ligase, and its targets, like SETB1. Capturing that moment where the destroy me tag is added is technically super challenging. Her meticulous work was vital for proving the erase the writer mechanism.
So real technical skill involved, too.
Absolutely. And remember, this wasn't just them looking specifically for an H3K9me3 regulator. It started as a broad, unbiased, curiosity-driven screen. They were just exploring basic biology, and bam, they hit on this major breakthrough. And this discovery, it really shifts how the field thinks about H3K9me3 stability. We used to focus almost entirely on the positive feedback, that snowball. Now we understand it's this delicate, active balance between writing the marks and erasing the writers. ASB7 is the key eraser. It really positions ASB7 as a kind of master rheostat for H3K9me3, like a dimmer switch, giving fine-tuned control. It's maybe comparable in importance to how another protein, USP7, regulates the famous tumor suppressor P53.
A master rheostat. I like that analogy. So not just on-off, but friendly adjustable. What are the potential practical implications then? If ASB7 is this control knob, could we maybe learn to turn that knob ourselves?
Yeah.
For therapies.
That's the exciting prospect. This definitely opens up completely new therapeutic avenues. Think about diseases where gene silencing goes wrong. In some cancers, for example, crucial tumor suppressor genes get silenced by too much H3K9me3, letting the cancer grow. Or maybe in developmental disorders, genes aren't silenced properly when they should be. ASB7 is now a potential target to address this.
How might that work?
Well, consider a really cutting-edge approach called PROTACs. These are basically engineered molecules designed to hijack the cell's own ubiquitin ligase system, exactly what ASB7 is part of, to target and destroy unwanted proteins.
Ah, so you could potentially design a PROTAC to use ASB7 to destroy a cancer-causing protein.
That's one possibility, leveraging ASB7's existing machinery. Or, perhaps more directly related to this finding, you could design molecules that modulate ASB7's own activity. Maybe you want to inhibit ASB7 in certain cancer cells to reactivate a silenced tumor suppressor gene by letting the writers, like SETB1, stick around. Or conversely, maybe you want to enhance ASB7 activity in a different context to reduce harmful gene expression by getting rid of the writers more efficiently.
So dialing the rheostat up or down depending on the disease.
Exactly. Now, of course, there are always caveats in basic research. This was done in specific cell lines, and there might be other related proteins, other ASB family members, that also play a role. We still need to explore that. But the core discovery provides this exciting new handle on epigenetic regulation. It gives us a novel way to think about tackling diseases driven by epigenetic imbalance. It's a shift from just thinking about the marks themselves to thinking about controlling the enzymes that write and erase them.
So we've gone from this image of an uncontrolled genetic wildfire of silencing to a much more nuanced picture, a finely tuned rheostat, ASB7, acting as this crucial brake, this dynamic regulator.
It's a really powerful reminder, isn't it, that even in biological systems that seem stable, there are these constant dynamic forces at play, keeping everything in this incredibly delicate balance.
Indeed. Yeah, this research really underscores that maintaining epigenetic stability isn't just a passive process of building things up. It's equally about actively taking things apart when needed. And it really makes you wonder, doesn't it? If ASB7 is this master rheostat for H3K9me3, what other hidden regulators are out there, quietly controlling other critical cellular processes, just waiting for us to find them?
Yeah.
And maybe, just maybe, learning how to manipulate these newly found brakes, these rheostats. Could it unlock entirely new ways to fight diseases? Could we potentially rewind or reset cells that have gone wrong epigenetically, bringing them back to a healthier state? It certainly opens up some fascinating possibilities.